So the next one is Sophie Hendrix again from the Hasselt University also, and talking about biosensors to monitor redox dynamics in plants.
So nice to see you all again this morning. So I already talked a little bit about flow cytometry on Tuesday, but today I want to talk to you about something else, which are genetically encoded biosensors. So as I told you on Tuesday, I did a postdoc in Germany, and that's where I really got a lot of experience with this technique. So the techniques that Anne just explained are all excellent techniques to investigate reactive oxygen species and different redox-related parameters in plants. But as she mentioned, the major disadvantage of this technique, at least depending on your experimental setup, is that there are steady state measurements. So it means you just measure at one time point. But now let's assume you expose a plant to cadmium and you want to know what's happening to ROS levels. If you want to know what's happening after 1 hour, after 2 hours, after 3 hours, you would have to harvest plants or stain plants every hour. And of course, that takes a lot of time. You need a lot of plant material and so on. And that's why these biosensors are very interesting, because they allow you to dynamically monitor what's happening to ROS levels, for example, over time. And that's what I want to show you today. So first, a brief overview of the things I want to talk about. So first of all, I will explain you, of course, what genetically encoded biosensors are. I will show you some advantages compared to the classical methods, if you can call them this way. Those are the ones that Anne just showed you. I will also tell you how you can classify these sensors and show you some examples. Then I will specifically talk about redox biosensors, because as you will see, there are also sensors for a lot of different parameters, such as calcium or ATP and so on. Then I will show you how to do the data analysis. And finally, I will finish with some results I obtained during my postdoc on heat stress. So first of all, what are genetically encoded biosensors? I will first start with the word biosensor. What does it actually mean? And in literature, I found this nice definition. So biosensors are molecules, organisms, or devices that are in a biological context, and they couple the sensing of a specific molecule or biological process that you're interested in to the emission of a quantifiable signal. Of course, sounds rather complicated. But what you could actually compare it to is, for example, a smoke detector. So a smoke detector will have a sensing mechanism that realizes, oh, there is smoke here, there is a fire. And then it will emit a quantifiable signal. So you know, then it starts beeping, there is a light flashing, and so on. And basically, that's what biosensors also do. So here you can see an example in a plant. So you have this sensory module, which really senses a difference, for example, in hydrogen peroxide levels, which is something you could be interested in. And then it also contains a reporter module, and this is usually a fluorescent protein. And this will then, both are coupled, and then you will get a measurable or quantifiable signal. So of course, not every sensor is the perfect sensor. I think none of the biosensors that exist today are perfect. They all have their drawbacks. But let's assume you want to design the perfect biosensor, then these are all things that you need to take into account. So first of all, the biosensor should, of course, be very selective for the analyte or biological process that you want to measure. For example, if you want to measure hydrogen peroxide, you want your sensor to only respond to hydrogen peroxide and not to other ROS, for example, or you don't want it to be affected by the pH or any other parameter. Then another important point is that it needs to enable a quantitative readout over biologically meaningful spatial, temporal, and concentration ranges. So if you want to measure changes in hydrogen peroxide in a plant leaf, then of course the sensor should be functional in plant leaves. Also temporal should be meaningful temporal. So if you want to measure for 24 hours, of course your sensor should be able to keep dynamically changing over this time period. And then something else that's very important is also the concentration range. If your sensor only responds to super high hydrogen peroxide levels, but you only have very low levels in your cells, then it will also not work. So those are all things to take into account. Then another characteristic of a perfect biosensor is that it has a high signal-to-noise ratio. So I told you usually these sensors work based on fluorescent proteins, but a problem that we have in many types of species, but especially in plants, is autofluorescence. Because plants, they contain chlorophyll, which is fluorescent by itself, and this can interfere with your analysis. So you want your sensor to be much more fluorescent compared to your background autofluorescence. And then the last very important point is that your sensor should not perturb the biological process that it measures or the biological system in which it is integrated. So we will really, as I'll show you in the next slide, integrate the sensor into our plants. But of course, you don't want the sensor to interfere with how a plant is responding to its environment, because that's what you actually want to measure. So you don't want to interfere with that. So we now know what biosensors are. Now, why are they called genetically encoded? Well, the reason is because we can really express these sensors in the plant genome. Now, how do we do this? We can do this via transformation via a bacterium called Agrobacterium tumefaciens. And what's special about this type of bacteria is that it can induce crown gall disease in plants. It's a type of disease which forms some sort of tumors. And this bacterium is able to do this because it contains here in its genome a plasmid, so part of circular DNA, which is called the TI plasmid. And at some point in history, some famous researchers had this idea like, what this bacterium can do is actually, through this plasmid, bring a little part of its DNA into the plant genome. So it can be stably expressed into this plant genome. Of course, this is not very interesting for the plant because it will get infected with this disease. But if now instead of this part of the DNA which it will induce the tumors, we put some other part of DNA which we are interested in, then we can actually use this method to transform plants with the DNA that we want. And that's actually how this works. So what you do is you take this plasmid, this TI plasmid, out of the bacteria. Then you cut the plasmid using a restriction enzyme. And then you can bring in basically any part of DNA that you want. So in this case, we will use a part of DNA that encodes the sensor that we want to use. And this is the purple part that you can see over here. Then you put it back into the bacteria, so you transform them with it. And then you infect the plants with the bacteria. And you can do this in different ways. And then in the end, you will have a transgenic plant, as we call it, because it contains some foreign DNA, where our inserted gene, so in this case, the gene coding for our sensor, is expressed. Now, after you generated these bacteria with the plasmid containing the part of DNA that you're interested in, how do you get it into the plant? So there's actually two main ways to do this. One is via leaf infiltration. And what you can then do is either spontaneously infiltrate the leaf. So you just put drops of the bacteria on the leaf surface, but this is super slow, so I don't think many people use this. But what you can do instead is use forced infiltration. So either you can use this syringe here, so you really push the bacteria into the veins of the leaf, or you could also use vacuum infiltration. So you put this leaf in a vacuum chamber and then the bacteria will basically be sucked into the leaf. What you will get in that case is a transient expression. So this is something that's usually done on tobacco plants or Nicotiana benthamiana. And then after, I think, 2 or 3 days, depending on the plant species, you will get the fluorescent signal in the leaves. But this is transient, so you can only measure it for a few days and then the signal will disappear. But what's even more interesting, but this is not possible on all plant species, is to use the floral dip method. And then you can get a stable expression. And this is something that's especially used a lot on Arabidopsis, so the model plant that we work on a lot. So if you have this Arabidopsis plant, and the image, it's not actually an Arabidopsis plant, but usually we use Arabidopsis. What you will do is you will have this solution with your bacteria containing the gene of interest, and you will just let your plant get to a stage where it starts to flower. So you have the little flowers there. And then, as the name of the method says, you dip the flowers into the solution. And then the bacterium will infect your plant cells. And then some of your cells will be transformed and will contain the gene or the protein that you're interested in. So in our case, this fluorescent biosensor. What you then do is you let your plant grow for several more weeks. At some point, they will start producing seeds. And then you need to select which seeds actually are the transgenic seeds, so which ones contain this protein that you inserted there. And you can either do that just by growing the seeds on some agar plates. And then in our case, because we're using a fluorescent protein, you can just very easily see under a microscope which of the plants are fluorescent. But you can, of course, also work, not in the case of biosensors, but you can also insert other types of DNA, which you cannot see under a microscope because they don't generate a fluorescent protein. But then you can also screen using antibiotic resistance, because this plasmid that we're inserting into the genome, it also contains an antibiotic resistance gene. So you can also select it in that way. So I will first briefly tell you something about why it's interesting to use these genetically encoded biosensors. And then I will also show you some examples and show you in more detail how they work. So the classical methods that just explained, they have some limitations, let's say. Of course, as she already mentioned, it's all depending on your experimental setup. If you only want to look at one point in time, maybe it's much easier to use these techniques, but it really depends on the question you want to answer. But these methods, they're often invasive. Sometimes you really need to prepare an extract, so you need to crush the entire plant or leaf, and then obviously you measure at one time point and you cannot use it anymore afterwards. But even the stains are quite invasive because you need to infiltrate them in the leaves and so on. They are steady state measurements, as I said, so you only measure at one point in time. But let's say you measure after 2 hours of cadmium exposure, then you still don't know what is happening to ROS levels after 1 hour or after 3 hours. So it would be more interesting to know what's happening over time. For some of these techniques, especially the ones where you need to prepare extracts, you also have a limited spatial resolution. Because if you prepare an extract from an entire leaf rosette, you see that ROS levels are going up. You still don't know, is it happening in all the leaves or only in a subset of leaves or in which part of the leaf? So that's another disadvantage. Of course, this is also time consuming because you need to prepare extracts and so on in some cases. And you also need a large amount of material. So, for example, the Amplex red method that Anne explained to you, if you have seedlings of Arabidopsis that are only 1 week old, they only weigh a few milligrams, but you cannot use that method for that. Then you really need to pool a lot of seedlings together to have sufficient weight to perform the technique. So that's, of course, a disadvantage. Now, the genetically encoded biosensors, because we can really insert them into the plant genome, they are non-invasive, so it means our plant will be fluorescent and we can just follow the changes in fluorescence over time without having to prepare an extract. We don't need to stain our plants with anything. We can just look at them under the microscope or in the plate reader, as I will show to you later. The major advantage of this technique is, of course, that you can do dynamic measurements, so you can really follow the fluorescence over time. You can do this at cellular resolution. So when you look with a microscope, you can really look in individual cell types, how the sensor is responding there. And you can even do it at subcellular resolution, because these sensors, as I showed you, you have to put the DNA coding for the sensor into your plant genome. But you can also add a targeting peptide, and this targeting peptide will make the plant cell know to which organelle the sensor needs to go. So you can really target the sensor, for example, to mitochondria, to chloroplasts, peroxisomes, the endoplasmic reticulum, and so on. Then, as I will show you later, both the actual measurement in the lab and the data analysis afterwards are largely automated. So once you generated these plants, you don't really need to do a lot. You just let the machine do all the work, and then afterwards for the data analysis, it's also quite automated. And you only need a small amount of material because you have your fluorescent plant here and you can just monitor the changes there, but you don't need tons of plants. Also, if you want to look over time, you're really measuring in the same plant over time. You don't need to harvest plants at all different time points that you're interested in. Now, yeah.
You don't need to take a leaf from the plant, so you sacrifice one leaf.
No, so it depends. Yeah, I will show also later in the analysis. So you can measure, for example, using confocal microscopy. And in that case, if you have a bigger plant, of course, you cannot put the whole plant under the microscope. So then you would indeed need to take a leaf or a root tip or whatever. But what I used to do is measure them in the plate reader, and then you can just put entire seedlings there. And then you're not even damaging the plant, let's say. So you just need to take it carefully with tweezers and you can keep it for much longer. So how can we now classify these sensors, or which types of sensors do you have? So I already told you that they work with this sensory module, which you have here on this side, and then coupled to a fluorescent protein. And then there's two options. So either you have sensors with one fluorescent protein, like the one that you see here. And how it then works is that you have a certain molecule that you're interested in. It's this red dot, let's say, is hydrogen peroxide, so a type of reactive oxygen species. What will happen is it will bind here to the sensory module. And then the sensory module will somehow induce a conformational change, so a change in the structure of the fluorescent protein. And because of that, the fluorescence will change. Now, there is also an option where you have two fluorescent proteins. I have to say for the redox sensors, it usually works with just one. But for example, sensors that we have in plants for calcium or for ATP, they work via this mechanism. So what you have then is, again, the sensory module here in the middle. Now our little red dot is calcium, for example. And here we have the two fluorescent proteins. And usually it's CFP and YFP. So CFP stands for cyan fluorescent protein. Cyan is some type of blue, so you can see it here. And then yellow fluorescent protein obviously fluoresces yellow. What will happen is when calcium is not present, so it's not bound, what you can do is you can use light at a wavelength that has the correct wavelength for excitation of the CFP. And then you will measure the emission and you will see that only CFP is emitting fluorescence. Now what happens if the calcium binds, as you can see here, then those two proteins come in much closer proximity to each other. And then instead of the CFP exciting light again, or emitting light, sorry, instead of the CFP emitting light, it will actually transfer this trapped energy to the yellow fluorescent protein. And then it's instead of the CFP, the YFP that will start emitting fluorescence. And this process of transferring the energy, it's called Fochter resonance energy transfer, or in brief, FRET. Now, how can you read out these changes? So I already told you there will be a change in fluorescence. So here for the single protein or with two proteins. And there are also two options or two types of sensors. Some sensors are intensiometric. So basically it means, just assume again an example with hydrogen peroxide. When more hydrogen peroxide is present, more of it will bind to your sensor and the fluorescence will go up. But you just measure with one excitation wavelength to excite the fluorescent molecule, and it will emit light at another wavelength. So that's the emission wavelength. So here, for example, you have all these curves, and you see the fluorescence emission intensity at the specific wavelength. And for example, if you have more hydrogen peroxide, your peak will move upwards. So you have a higher signal. The problem with this is that your signal, as we want, it will be affected by the concentration of hydrogen peroxide, but it will also be affected, for example, by stress factors. So I worked in my postdoc with heat stress. What can happen is, of course, heat stress, especially if you use very high temperatures, it will start damaging your fluorescent protein. And then, of course, the signal will go down, but the signal is not going down because you have less hydrogen peroxide. The signal goes down because your sensor is simply damaged, and you don't really know this. So that's why it's quite dangerous to use these intensiometric sensors. So you should always be careful, with interpreting the data you obtain. What is actually much more interesting, and we try to always develop these types of sensors, is to use ratiometric sensors. And what will happen in this case is that either you have these two fluorescent proteins or one fluorescent protein, but you make a ratio of fluorescence either at two excitation wavelengths or two emission wavelengths. And maybe here it's not super clear, but I will show you an example later on where I think it will be more clear. But here, for example, you have a sensor which will always emit light at the same wavelength, but it has two excitation wavelengths. And depending on whether your sensor is oxidized or reduced, which depends then on the hydrogen peroxide that is present, it has a different excitation spectrum. So here you can see that it has an excitation peak around 550 nanometers and a second peak around 420 nanometers, I would say. And here, for example, this light red curve, you will see here it has a small bump. Here it has a much bigger bump. But then if you look at this darker curve, then the peak is higher here and lower here. So this means if one excitation drops, the other one increases. And what you will do then is in your readout, you will make a ratio of both. Of course, it's still possible that heat stress, for example, damages your sensor. The protein will get damaged, so the fluorescence will go down. But then the fluorescence of both peaks will go down, and the ratio between both will still give you a reliable readout of hydrogen peroxide levels, for example. So these are much safer to use because they're not influenced by the amount of sensor that is present. Now, I already told you we have a lot of types of genetically encoded biosensors. Today, I'm only talking about plants, but you need to know you can also use these in animals and bacteria and cell cultures and so on. So this is something that, if you're working with another organism, you can definitely look up some research and you will for sure find something. I don't know if you can use it in the bears, but fluorescent bear might be a little bit strange, but especially with cell cultures, for example, or flatworms and so on, this has been used as well. But in plants, we already have quite a lot of these sensors. So for example, we have sensors for pH, for sugars such as glucose. We have a sensor for magnesium ATP, which is the form of ATP that enzymes use. There is a lot of sensors for calcium, also sensors for phytohormones, so plant hormones such as abscisic acid, gibberellic acid, but we also have sensors for nutrients such as phosphate or zinc. But the ones that I will talk to you more about today are the sensors for redox-related molecules. So here you can see an overview of all redox sensors that exist in plants. So you can see that there are quite a lot. There is only one of them here that works via this FRET mechanism that I showed you with the CFP and the YFP, so the two fluorescent proteins, but I will not talk about that one today. It's a sensor for thyrodoxins, which are a certain type of antioxidants, antioxidative enzymes. But most other sensors, they work with only one fluorescent protein. And then we have sensors for hydrogen peroxide, which we already talked a lot about. We have sensors for the ratio between NADH and NAD+, which is, of course, an important electron donor and therefore also strongly linked to redox reactions. And we also have sensors for the glutathione redox potential. So as Anne mentioned, glutathione is an important antioxidative metabolite. And if it gets more oxidized, it will also tell us something about the redox state in our plant cells. So I will not explain all of these to you today because it's really a lot, but I just selected a few. And here, for example, we have the hypersensors, which are very popular sensors for hydrogen peroxides. And how do they work? So as I mentioned, they only have one fluorescent protein. And this is the CPYFP, so it stands for circularly permuted YFP. So it's just a yellow fluorescent protein, but they made some changes to it. That's why it's called CPYFP. So this is our reporter domain. So this will give you a signal. Now, how do we make it specific for hydrogen peroxide? Well, that's by coupling it to this part here. And this is called the OxyR sensing domain. And this is a part that they took from a transcription factor that's present in bacteria and that they know is very responsive to hydrogen peroxide. So what will happen is when hydrogen peroxide is present, it will oxidize this domain over here. And as you see, the domain contains two thiol groups, so it's an SH group. And what's typical for these thiol groups is when they get oxidized, they will form a disulfide bridge. So you get this bond here between the two sulfur atoms. And what this does is it causes a conformational change. So the protein will change its conformation a little bit. And as a consequence, the excitation spectrum will change. So here we have the reduced molecule. Here we have the oxidized molecule with the disulfide bridge. And they will both have the same emission wavelength. So they always emit yellow light. However, the light that you need to use to excite it, so to get it to this higher energy state, that's different for the reduced and the oxidized sensor. So here on the y-axis, you always see the fluorescence intensity, so the fluorescence emission at, I think it's 530 nanometers. And here you see on the x-axis, the excitation wavelength that was used to obtain the fluorescence emission. And then here you can see the spectrum for the reduced sensor in black. So you see that there is a peak here around 500 nanometers and a second peak around 420 nanometers. Now, if the sensor gets oxidized because hydrogen peroxide is present, you get this disulfide bridge, you get a conformational change. And then you will see that the spectrum will completely shift because this peak here around 420 nanometers, it completely disappears. And you get a much higher peak here around 500 nanometers. And because we have these two excitation peaks, this is a ratiometric sensor, as I just showed you. Now, in plants, there are different variants of this hyper use. In animals, you even have much more. But the first one was just simply called hyper for hydrogen peroxide. But the big disadvantage of this one is that it's also pH sensitive. So if you see a change in the spectrum, it could mean that there is a difference in hydrogen peroxide levels, but it could also mean that the pH in the cells is changing. And of course, that's not what you're interested in. So to control for that, you need to use plants with an extra sensor, which is called Cypher. And this will only respond to pH, but not to hydrogen peroxide. And then you should check, does the hyper change? Does the Cypher change? And based on that, you can draw a conclusion on whether you're actually measuring changes in pH or in hydrogen peroxide levels. But of course, this was the one that was used really in the beginning. In the meantime, people have worked on engineering these sensors, making them more and more specific for hydrogen peroxide. And very recently, a novel sensor has been introduced in plants. And this one is called Hyper 7. We don't have the other 5 in between here in plants, but in animals, they do exist. But this Hyper 7, they also put in plants. And what's nice is that it has a much lower pH sensitivity. So if you change the pH in a reasonable range, it will not affect the sensor. So it's only sensitive to hydrogen peroxide. Then two other sensors that are interesting. They are both based on RoGFP2. So the fluorescent protein here is called RoGFP2. It stands for reduction oxidation, and then GFP is green fluorescent protein. This one here, it's coupled to ORP1. And this sensor is also specific to hydrogen peroxide. So it's another one that you can use instead of hyper. And this one here, it's coupled to something else that I will show you here, which is a human glutaredoxin. So this sensor we call GRX1 RoGFP2. And this one doesn't, it also responds to hydrogen peroxide, but not directly. It actually responds to changes in the redox potential of glutathione. So remember, glutathione is an important antioxidant. And when the conditions in the cell become more oxidizing, then glutathione will be oxidized to glutathione disulfide. So it's basically also two glutathione molecules with this disulfide bridge in between. And this sensor specifically responds to changes in this redox state of glutathione. So what we have here is the glutaredoxin 1. And when GSSG, so the oxidized glutathione, is present, it will get coupled here to one of the style groups of the GRX1. Then this is transferred to the fluorescent protein, which is our RoGFP2. And then in the end, what will happen here is that a disulfide bridge is formed between these two tile groups of the RoGFP2. And this will again cause a conformational change in our fluorescent protein. And very similar to what we saw for the hyper, it will cause a change in the excitation spectrum. So the emission will still be at the same wavelength, but the excitation will change. Now it's the opposite as for the hyper. So for the hyper, we saw that we had 2 peaks for the reduced version, 1 peak for the oxidized version. Here it's the other way around. So we see 2 peaks in case our sensor is more oxidized. So a peak around 490 nanometers and a second peak here around 390 nanometers. And then with the reduced sensor, we only get this second peak at the higher wavelength. So now you more or less know, or at least I hope you now understand how these sensors work. Now the question is, how do you really measure this? So you generate your plans expressing the sensor, but what do you do now? If you go into the lab, you want to measure the responses, how do you start? So basically what you need to do in your plans expressing the sensor, and here I will use the GRX1 RoGFP2 for the glutathione that I just showed you. You will always measure the fluorescence emission at 530 nanometers because that's at a fixed wavelength. But you do this one time after excitation at the 1 excitation wavelength. So here it's 410 nanometers. And a second time after excitation at 480 nanometers. Now, to give you a very easy example, let's assume we expose our plants to hydrogen peroxide. What we expect then is that the glutathione will get oxidized to glutathione disulfide. And that's what we did over here. So here I put again the spectrum just so you can remember what it looked like. So here we had the reduced sensor in blue, the oxidized sensor in red. So what we expect is if we give the plants hydrogen peroxide, we will get more of the GSSG. So the sensor should become more oxidized. And that's also what we see over here, because here we add the hydrogen peroxide and then we see that there is an increase in fluorescence after excitation with 410 nanometers. And at the same time, you see a decrease in fluorescence after excitation with 480 nanometer. So this is what it looks like. And this is what we call a ratiometric response, because one fluorescent channel goes up and the other one goes down. Then after some time, you see that they start coming back together. That's simply because the plant cell is actively investing into detoxifying this hydrogen peroxide, because as nicely explained, if you get a lot of this hydrogen peroxide, it causes damage. So you want to, of course, avoid that. Now, what do we do afterwards? So after we performed excitation at 410 and 480 nanometers, we 2 times again measured the emission at 530 nanometers. And from these emission values, after excitation at both wavelengths, you calculate a ratio. So you calculate the ratio between the emission after excitation at 410 and emission after excitation at 480. So you just divide them by each other. And then instead of having these 2 values, you combine them into 1 value. And then the responses will look something like this. And here we have curves showing responses of the sensor in plants treated with different concentrations of hydrogen peroxide. So you have here a concentration of 0 millimolar, so they were not treated. Those are our controls. And when you follow then this ratio over time, you see that it's a flat green line because nothing is happening, which is what we expect. Then if you give them a bit of hydrogen peroxide, 1 millimolar, you see that you get this little peak here. And then over time, it goes back down to control levels because the hydrogen peroxide gets detoxified and also glutathione gets reduced again. And you see with higher and higher concentrations of hydrogen peroxide, the sensor gets more and more oxidized, but it always goes back to control levels. And here you can really measure at many points in time in the same plant, so you just really dynamically monitor this. And then, especially for these redox sensors, it's also interesting to perform so-called calibrations. And what that means is that you have some additional plants that you treat either with a lot of hydrogen peroxide, so a very high concentration, or with a lot of DTT. DTT will reduce the sensor. And then you have an idea of the dynamic range of the sensor. So that means which values do you get when the sensor is completely reduced? Which values do you get when the sensor is completely oxidized? Because, for example, if I work with heat stress and I see an increase in this ratio, I want to know, is this now a very strong increase or is it mild? And you can do that by doing these calibrations. And then you just get these dotted lines at the top and the bottom of the graph, which show you which is basically the range in which the sensor can respond. So you see here, if this peak goes up completely to this top line, you know that the sensor is really completely oxidized. So that's a really strong stress condition. Yeah. Yeah. And it's basically some type of control that you're doing indeed. And you also have two options. So either you use separate plants to perform them on, or you can have your plants that you treat with heat stress or with cadmium. And then at the end of your experiment, you treat them with hydrogen peroxide first. So you should see a very big peak. And then you treat them with DTT, and then it should go down again. And then you also know really in the same plants with the dynamic ranges. But usually we use a combination of both. Yeah. So yeah, now I already told you how this data analysis works. So what should be very clear is that you need to measure fluorescence. Now, which device will you use to do this? There you actually have two major options. The first one is to use confocal laser scanning microscopy. And this has, some advantages because you can really look at individual cells, which could be of interest depending on your research question. And here you work with small tissue sections because, as Ingeborg also asked, a microscope, you cannot put an entire plant underneath it. So you need to use, for example, an individual leaf or a root tip or just smaller parts of the plant. You look at the cellular resolution, so you can really check in which cell types you are seeing a response. It's relatively low throughput because you can only put one root tip under the microscope at a time. So you really have to check each tissue one by one. And it's also for relatively short monitoring durations because, of course, you don't want to sit at the microscope for 24 hours in a row to gain your results. So how would this look? Well, here is a system that they were using in Germany. So here, this is where you put your plant and it will look something like this. So here, this thing in the middle is your Arabidopsis seedling. And then you have a system connected to it where you can just pump buffer over the seedling. And with this buffer, you can mix, for example, hydrogen peroxide, DTT, but also some other compound that you could be interested in, like cadmium or whatever you want to study. And then you can just follow the changes in fluorescence in this seedling over time. And here you can see this ratio that we just talked about for two different sensors. So this is the sensor GRX1 rho GFP2 for the glutathione redox potential. And here you have the rho GFP2 ORB1 in lighter gray, which is another sensor for hydrogen peroxide. And here you see first we put just regular buffer over the seedling, and then our ratio is very stable, which is nice, of course. Then when you add hydrogen peroxide, you will see that the ratio goes up. Then you add buffer again and it will stay stable for some time, but then the sensor starts to spontaneously reduce again because hydrogen peroxide is being detoxified by the antioxidant system. But then when you add DTT to reduce the sensor, you will also see that it goes down again very rapidly, back to control levels. So this is just a ratio plotted in a graph, but you can also really show the images. And this is what we call false coloring images, where a blue color stands for a fully reduced sensor, and a more reddish color stands for a fully oxidized sensor. And then here, basically, you can see the same thing that you see in the graph here. So the sensor goes from reduced after treatment with hydrogen peroxide, it gets more oxidized, so it starts becoming greenish, even completely red here, and then it starts to reduce again until it's fully reduced. So we said we can use confocal microscopy, but it has some limitations, also some advantages. But another technique that you could use is to study or to measure the fluorescence in a plate reader. And this has some advantages. So here you can use larger tissue sections. So you could use entire young seedlings, so the small plants, and you have the roots and the leaves, everything still intact. Or if you have bigger plants, which are already much older, then you can also use leaf discs. So you just with a hole punch, push out some discs of the leaves. Or you could detach the roots, put those into a well, for example. A disadvantage here is that you don't have the cellular resolution. If you're measuring in an entire seedling, you will just get one fluorescence value for the entire seedling. So you don't know, is the response mainly occurring in roots and leaves? Is it both? So that's something you're missing in this case. But what's the most interesting about this is that it's super high throughput because you have 96 well plates. So in each well, you can basically put plants and measure something else. You could put a different cadmium concentration in each well, or you can just test a lot of things simultaneously. And what's also nice is that you can monitor for quite long durations, let's say up to 1 day. And it's really not time consuming at all. You just put your plants in the wells, you put it in the machine, and you can tell the machine, I want you to measure the fluorescence every minute, every 2 minutes, every hour, or even every few seconds. So you can have as many data points as you like, basically. And you don't have to do anything, just sit and wait until the machine is done. So this is what the plate reader looks like that we use for this. So here you have a 96 well plate. It's not super clear, but here at the top, I have some detached roots from plants grown in hydroponics. So they're 3 weeks old, more or less. Here I have some leaf discs that I cut out from larger leaves. But you could also use smaller seedlings. So then you have the entire plant. So here there is this animation, but in real life, it looks like this. So what you do then is you just put several seedlings in the well. And then what the plate reader will do for you is every time it will perform excitation at 410, measure emission at 530, and do the same thing then after excitation with 480. Yeah. Yeah. So you could try to do it with one seedling if they're quite big, but the only problem then is, you see here in this example, the wells are quite nicely covered by the seedling. And this system here, this plate reader that we use, it uses a system called orbital averaging. So if you just have this entire well, it will measure the fluorescence at different spots inside the well. But of course, if you have this one tiny seedling that's stuck to the side of the well, then it won't measure much fluorescence. So it's always good, yeah, to fill the well relatively, until it's relatively full. Of course, you also don't want to put 20 seedlings in there because then the plants will just get stressed from being squeezed. But it's always good to have several just to, yeah, get the best possible readout. And these are then the seedlings inside the plate reader. Here we have on the left side a wild type plant, so it doesn't have the sensor. And you see that when you check for the GFP signal, it's not fluorescent. Here you have the sensor with, here you have the plants with the sensor, and you see when you check them under a microscope, for example, that they're nicely fluorescent. And I didn't show you that for the data analysis today, but because our plants are also autofluorescents, you'll also always measure some fluorescence in plants without the sensor. And we use these as some sort of control. So in our calculations, we also use some samples or plants without a sensor to correct for this autofluorescence. Now, if you want to do this analysis in the plate reader, just a short overview of what you would really need to do in the lab. So you first grow your plants for 7 days or less, 1 week, on agar plates. Then first you check the fluorescence under a microscope, but just very generally for all the plants on your plate. The reason why it's good to do this is because sometimes these sensors that you express in the plants, they get silenced. So somehow the plant feels like, why do I even have this sensor? I don't need it for anything. And then they silence it. So they're no longer expressing it. And that's, of course, something you want to avoid. So usually on a plate with 50 seedlings, you have like 5 or something that are very dim, and then we just leave them out. Then the seedlings that are nicely fluorescent, you put them in this 96 well plate. Here I put 5 seedlings per well, but it depends a bit on their size. If they're small, you put some more. If they're very big, you put less. And we put imaging buffer, just something to keep the pH stable, solution that the plants like to be in. Then you transfer this plate into the plate reader. And then first you measure the fluorescence without applying any stress conditions, just to see whether it's stable. So you want your ratio to just be a flat line, ideally. And then you can apply any type of stress that you're interested in. For example, cadmium, yeah, you can apply it as a solution. This plate reader that we were using, it even has some automatic pumps. So you can just put a small tube with cadmium solution there, and you tell the machine after 1 hour, you inject this volume in all of these wells. Another thing that I'm interested in is heat stress. What you can do then is just increase the internal temperature of the plate reader to induce the heat stress. And then you will just kinetically monitor the responses over time. And to finish, I will show you some examples of the work I did with heat stress in Germany. So here I performed one experiment with, let's say, more mild heat stress. So here, this is just the experimental setup. So my control plants, you always need some controls. I just kept at a temperature of 25 degrees continuously. And then the stressed plants, I first kept at 25 degrees for 1 hour to measure this baseline fluorescence. And then I increased the temperature in the plate reader to 37 degrees. And then here you can see the response of this GRX1 RoGFP2 sensor for the glutathione redox potential. So this first hour for the baseline, it's in front of this dashed line. You can see not that much is happening. And then my control plants don't show any change in fluorescence. But my heat stress exposed plants, they first show a small oxidation of the sensor. And then around 6 or 7 hours into the experiment, they get fully oxidized. So you really see that the heat stress very strongly oxidizes the glutathione in the plant cells. Then on the right side, we have the Hyper 7 sensor. So this is one for hydrogen peroxide. And what you can see then here is actually a very similar response. So here a very tiny oxidation, and then towards the end, you get this massive oxidation. So then at that point, the plants are really not happy anymore. Then I did the same thing, but then with a more severe heat stress. So this time I used a temperature of 43 degrees instead of 37 degrees. And what I saw then is actually very similar responses. But what you need to keep in mind is here my time frame was 22 hours. Here it's only 8 hours. So the response looks very similar, but it happens much faster. So here you see the full oxidation only after 10 hours. Here you already start seeing it after 3 or 4 hours. So the plants respond much faster to this higher temperature. And then even with the Hyper 7 sensor, here the responses started looking very strange after 4 hours. So I only included the first 4 hours of the experiment, because as I told you, you also need to keep in mind these sensors, yeah, they're proteins, they can also be damaged by heat. So at some point, yeah, the sensor will just be damaged and it won't properly respond anymore. But at least for the first 4 hours, it was very clear that as soon as I started to heat stress, immediately the sensor started to oxidize. So it means the plants are really producing lots of hydrogen peroxide. And then just the last slide to show you that really these sensors allow you to dynamically monitor changes. What I did here is a recovery experiment. So I again brought the plants to 43 degrees for 1 hour. But then I reduced the temperature back to 25 degrees to see whether the plants would be able to recover. And you especially saw this very nicely with the Hyper 7 sensor. So as soon as I increased the temperature, the sensor started to oxidize. But as soon as I decreased the temperature again, the sensor started to get reduced again. So you can really follow these changes over time in the same plants. And what was quite interesting is for this glutathione biosensor, I didn't see anything in this first hour, which is what I expected. But then I even saw that the sensor got more reduced. And this is, I think, some sort of overcompensation mechanism, because the plants think like, oh, I'm being stressed, glutathione is going to get oxidized. And then it can activate some enzymes that are responsible for keeping glutathione in its reduced state. So it would even get more reduced than it would be under control conditions. So this upper part of this slide I already showed you, so the advantages of these sensors. Yeah, I hope that I could convince you that they are very interesting to use. But what you do need to keep into account is that they also have some limitations, because these classical methods that Anne talked about, you can basically use them in all plant species. If you have a new mutant that you're working with, you start working with it today, basically you can do this DOP staining or amplex red staining tomorrow. If you want to do the same thing with the genetically encoded biosensors, you need to take into account that you still need to transform plants with the sensor. As long as you're working with wild-type plants, it's fine because we have them with the sensor. But let's assume you have a new mutant that you want to start working with. That means you would have to start transforming it. But first you need to grow it until it's flowering, then you can transform it. But then you still need to select the fluorescent seeds, go to the next generation, and so on. So it can easily take up half a year or even longer to have the material ready to start working with it. So that's something you need to take into account, so that you need to transform your plants. And also this is not possible in all plant species. So this floral dip method that I showed you works very well on Arabidopsis. Also this transient expression using infiltration, you can use it on some plant species, but it won't work for all of them. So if you're working with more of a crop species, it's probably at this point at least still best to go for these classical methods. But I do think in the future, more and more of these advances will be made so that these sensors will also be used more and more in crops and economically relevant plant species. So then just a last slide with a few take-home messages. So what you should remember is that genetically encoded redox biosensors, they consist of a sensory module coupled to either one or more fluorescent proteins. They can either be transiently or stably expressed in the plant genome, plant between brackets, because you can also use it in other species. They allow dynamic monitoring of a parameter of interest. And here the dynamic part is super interesting because that's really the big difference compared to the other methods. They can be targeted to different subcellular compartments, which is very interesting if you want to look, for example, specifically at responses in chloroplasts or the ER and so on. And they're, of course, excellent tools to study stress-induced effects on redox homeostasis.